Some of the most advanced applications of pharmacogenetic knowledge are those in the field of cancer therapeutics. Cancer is an unusual disease as it arises through the accumulation of multiple mutations in certain genes within a cell that together act to disrupt the normal checks and controls over cellular function and replication. This leads to uncontrolled and inappropriate cellular proliferation. Cancer cells therefore have distinct genetic profiles. Increasing understanding of the molecular genetics of cancer is making it possible to exploit the differences between normal and cancerous cells to target therapeutics. The major problem with conventional chemotherapy is that to achieve reasonable efficacy, a substantial degree of toxicity, and hence significant adverse side effects, is required. Targeting drugs or other therapeutics more specifically to tumour cells both increases efficacy and reduces toxicity. One of the best-known examples of genetic targeting is that of Herceptin. This is an antibody-based biological therapeutic used for the treatment of HER2-positive breast cancers, that is, those that produce abnormally high levels of human epidermal growth factor, about 20% of all breast cancers. Breast cancer cells that produce high levels of HER2, a cell surface molecule, are stimulated to replicate by the binding of HER2. Herceptin competitively binds to HER2, blocking growth factor binding and targeting the cancer cells for destruction by the body's own systems. Non-cancerous cells do not produce large amounts of HER2 and are relatively ineffective, reducing toxicity of the treatment. Purists might argue that the design of therapeutics such as Herceptin and Gleevec do not represent true pharmacogenetics because they are specifically targeted to the genetic nature of the tumour cells as opposed to the patient's genotype. But the commercial success of these agents has had a significant impact on the profile of pharmacogenetics as a tool for drug design and development.